Okay. Hi guys, good morning everybody. Today is my pleasure to introduce Isaac Cervantes-Sandoval. He's an assistant professor at the Department of Biology in Georgetown University. A little bit on Isaac's trajectory. He's a graduate of the National Polytechnic Institute in Mexico City from the Escuela Nacional de Ciencias Biológicas. I don't know quite yet how to translate that in a short note. He's a biologist, right, Isaac?
Yes.
Actually, we met there
Yeah.
During college. Then he moved to perform a master thesis and PhD degrees at the CIMBESTAF also in Mexico City. He was studying the role of innate immune response in pathogenesis of amoeba meningoencephalitis under the supervision of Dr. Mineko, that sadly passed away recently. Then he moved to perform a postdoctoral position at the Department of Neuroscience in the Scripps Research Institute, where he was also later promoted to senior staff scientist because he's awesome, like he's very productive scientist. And very recently in 2019, he started his assistantship professorship at Georgetown University in the Department of Biology. So Isaac is very interested in understanding memory, and he used fly as a model, as you may have seen in the ad. And today we're very fortunate to have you here, Isaac, sharing your research with us. So with no longer introduction, please, we're all yours. Thank you.
Thank you, Teresa. Well, thank you everybody for coming. Yeah, I met Teresa a very long time ago when we were in college. We actually went. I don't know if we ever took the same classes in the same classroom, but
no,
we
you were in
have a friendship relationship, and then we also met a few times when we were at CIMBESTAF.
Yes.
So that was pretty cool, and then we kept contact. So we are good friends, and she has been very helpful in all this. Having come from Mexico and been moved towards the steps here in the US has been really tough, challenging. And then Teresa, she has always been there for me and helping me. She helped crafting my cover letters when I was applying for a job. So she's been very helpful all the time. So I'm very grateful to her. Okay, so today I'm going to talk about a little bit about my research at Georgetown. So my lab is very interested in trying to understand how memories are encoded, how memories are consolidated into long-term memories. So usually when we learn something, if this thing that we learn or we experience is not very relevant, we tend to forget that. We tend to, just get rid of those memories. But there are some memories that are transformed into long-term memories, and then they can last for a very long time, days, months, and even a lifetime. But also, most recently, we are very interested in trying to understand how do we forget and why do we forget. And so we tend to forget. We tend to think that forgetting is something undesirable, and that's because we usually associate forgetting with some pathological conditions like Alzheimer's disease or dementia and things like that. Also, forgetting something that is really important can get you into trouble. And I know this because I'm very hard forgetful person, and that has caused me many problems in life. So, so many things I find myself thinking, wouldn't it be great if I could remember everything, if my memories were like perfect representations of the past. But then when I start to think deeply into this, I start to realize that maybe forgetting is not such a bad idea. And this is very well depicted in a fictional character from Jorge Luis Borges. This guy is called Funes, and he was a normal being, and then he had an accident. And after the accident, he could not forget anything. His memories were, like I said, like they were perfect representation of the past. They were like video recordings. But with this perfect memory came a lot of cognitive problems. He couldn't function normally because everything that he was seeing or experiencing will trigger memories in his mind, and then he will spend all his time just remembering things and couldn't experience anything else. For him, remember one day of his life was it would take him one full day. So it was very clear that there was a lot of cognitive problems and that arise from this inability to forget things. So intuitively, we can think that forgetting is important in at least 4 different ways. If we think that our brain has a limited capacity, like a memory from a computer, for example, and then we do experience a lot of things in our life. So we every day we, walk in the street and we see things, and there's a lot of sensory input. And if we were storing all this information, well, you can think that this will fill our memory capacity really fast, and then we won't have any more free space to store new things. But that doesn't seem to happen. It seems like we always have some more space to store new information, relevant information. So we think that forgetting is important for avoiding saturation of the brain capacity. So basically freeing space from our brain. Another thing that we think forgetting is important is that it increases the efficacy during memory recall, okay? And so imagine that this desktop is your memory system and all these files are just previous memories. Now, think that you want to retrieve a memory from all this mess. It will take you a very long time to find the right memory, and it's just not gonna be any efficient at all. Now, imagine the different scenario where you, instead of storing all these files, you just deleted the ones you don't need anymore and just kept the ones that are important in a better organized manner. So now, if you want to retrieve a memory from here, it's going to be way easier and way faster. And so people think that forgetting is really important for this memory recall efficacy. One more thing, we live in a very dynamic environment, changing environment, right? Animals live in a very changing environment. And so we need to maintain our behavioral flexibility. Now, imagine that you have a favorite restaurant, and this is your home, this is your restaurant, and you know exactly where it is and what's the pathway that you have to take to get there. Now, imagine they relocate this restaurant. Well, you will learn this new location, and with time, you are going just to forget the old location. And in this way, you just update your memory system to things that are changing. And we believe that forgetting is important to keeping this memory flexibility. One final thing, forgetting is really important for memory generalization. Now, let me put this as an example. Imagine that you're a mouse and you have encountered many cats in your life, and you know they're dangerous because they try to eat you. Now, each one of these cats is represented here in this graph by a blue dot, and its position in the graph is based on its characteristics, right? Like the smell, the color, the size, whatever you want. Now imagine your memory is a perfect representation of the past, and then you create a model to predict what a cat is or what a feline is. If your model is a perfect representation, and then it will look more like this black line, which is going to be very fit, is going to cover almost every point on the graph. But now let's imagine you find a different feline. Now this is a bobcat, and this is represented by this green dot. If you have a overfit model like the one on the left, you will fail to predict that this is also a feline and it's also a predator that can kill you. Now, by means of generalization, what generalization means is you forget about the little details of each specific cat, like the color or something, and just remember the gist of what a feline is. Now, your model is going to be something like this black line in the right. and it's going to be better to predict what that this feline is also dangerous. This process of memory generalization is really important for survival or for cognitive performance. And forgetting is fundamental for this to happen. Okay, so this is what we think forgetting is doing in normal forgetting, not pathological forgetting. And we are trying to understand how this is happening in the brain. For that, we work with fruit flies, so Drosophila melanogaster, and this is the way we train the animals to do memory assays. So we put a bunch of flies in an empty tube like this, and then we can pass an odorant and pair that odorant with a shock. And then we wait 30 seconds where they are getting just clean air, and then we presented a second odor, different odor. And this time, this is not associated with the shock. Now, after training, we put the flies in a T-maze that look like this, and we can pull the two odors that we use during the training through the two different arms in the maze. And we ask the flies to decide for 2 minutes. Now, if the flies remember the task, they will avoid the odor that was associated with the shock and then move towards the other odorant. And in this way, we can score how well they remember. Now, this is a Drosophila brain. This is a picture that, from a confocal microscope. And this is just a cartoon of how it looks. Now, this is an olfactory condition. So that means that olfactory information gets to the brain through the antenna. So this is like the Drosophila nose. And it gets to these olfactory antenna lobes, which they are here, these green things. And the neurons from here, they project to the back of the brain and synapse to a very large group of neurons called margin body cells or canyon cells that are in the back of the brain. Now, these canyon cells send projection to the front, axonal projections to the front, and they bifurcate, and they form these L-shaped structures, which are called motion bodies. Okay? So this is a model of the circuit that is important for olfactory memory encoding. So these L-shaped structures are formed by these canyon cells, or these motion body cells. And these cells have, of course, olfactory input, but they also receive dopaminergic input from two types of dopaminergic neurons. So they have these red ones called PPL1 that innervate basically the vertical lobe. And then we have some other dopaminergic neurons that called PAM neurons, and they innervate mostly the horizontal lobe. Now, by a very large series of studies, we know that PPL1 neurons convey punishment signals during aversive olfactory conditioning. So the one that I just told you about. And PAM neurons, they send reward information during appetitive conditioning. So we can also train the flies to associate odors with sugar rewards, for example. And those signals, so those rewarding signals are conveyed to the motion bodies by these PAM neurons. There's a third part of the circuit that is really important. These are called very creatively motion body output neurons. So these neurons are basically taking the information from canyon cell, and then they project to different parts of the brain, and they basically drive the behavior. And we know this because if we optogenetically activate this branch, for example, the blue one here in the vertical lobe, this activation drives approach behavior. So they like it, flies like it. So flies will tend to approach to that light that we are using to stimulate. And conversely, if we stimulate this other branch, the one that innervates mostly the horizontal lobe, that drives avoidance. So they don't like it. So you can see how they kind of are inverted. So the punishment signal comes here, and then the output from this, let's say this part, it drives approach, and then the opposite happens in the horizontal lobe. Now, this is what we think is happening during learning. So during aversive conditioning, for example, there is canyon cells receive olfactory input. In this case, only few neurons from the thousands of canyon cells will respond to specific odor, and that's represented here by this darker cell. So this is the neuron that is activated by a particular odor. And then during training, we are basically delivering electric shock. So these dopaminergic neurons are activated, and there is going to be a coincidence of the two stimuli during aversive olfactory conditioning. So right here at this node, they are receiving both odor and shock information. And that is going basically to depress that connection, okay? That synaptic connection is going to get depressed. And so what is going to happen is that during memory retrieval, when you present the same odorant to the fly, the same canyon cells are activated, and then they send the activity towards the axonal projection, and then it basically inputs both of the immersion body output neurons. But now, because this node is depressed, the approach output is reduced, and now the avoidance is basically you have tilted the relationship between these two branches, and now the end result is that you have an avoidance behavior. Okay, so this is what we think is happening and how memories are being encoded. Now the opposite happens with appetitive conditioning, and it's just the other branch. In this case, this is the node that gets depressed, and now the approach immersion body output neurons have a relative higher weight, and then drives approach. When we train the flies with this aversive conditioning, we usually get most of the flies avoid the odor that was used for training. So this is just a measurement of the memory. It's called performance index. And a score of 1 means that all the flies remember the task. A score of 0 means that no fly remember. And so if we test the memory right after training, flies remember very well. But then with time, they start to forget. And then by 24 hours, most of the flies don't remember anything. Now, just like in mammals and humans and any other animal model that we know, if we repeated the training phase, so if we train the animals several times with some resting interval between each training session, they remember better. So they get consolidation, memory consolidation, and they form these long-term memories that are protein synthesis dependent. And so 24 hours later, they remember much better than the ones that receive one single training. And actually, this is 24 hours, but if you look at 2, 3, 7 days, this training with 5 repetitions will last for even weeks. But the one with one will just forget. So we are trying to understand how is this happening? How is this forgetting happening? Now, all of these studies started more or less in 2012 about forgetting. And I'm just gonna give you a summary of what we found. This was reported in 2012, and we actually found that the same dopaminergic neurons that convey the shock information during learning, they remain active after learning. So after learning phase finished, these neurons actually keep releasing dopamine. And this dopamine, it's activates this dopaminergic receptor, and that actually induces this memory forgetting. So same neurons that were used for the learning phase, they just keep firing at a lower rate, of course, and then that conveys what we call a forgetting signal, and that induces the memory decay. Then in 2015, we found that this signal is actually dependent on the behavioral state of the animal. So if the animal is active and moving and flying around, these dopaminergic neurons are very active. They have a lot of activity going on, and dopamine release, and that induces that forgetting that we see after learning. But if the flies are resting, they're quiet, then this dopaminergic signal is low, and that impairs forgetting. And we can manipulate this actually. So we know the sleeping circuit in the fly. We know what neurons are important for sleeping, and then we can activate them artificially and basically put the fly to sleep. And we see this dopaminergic reduction, activity reduction, and we see an impaired forgetting. So basically, this is what we found. Dopaminergic activity is important for plasticity, and the behavioral state of the animal is going to determine how much activity there is in these dopaminergic neurons, and that's going to drive this forgetting signal. Okay. But we really wanted to understand a little bit more about the molecular mechanisms of how forgetting is happening. And so we perform a huge screen. We screen thousands of different RNAi lines and look for genes that were important for this memory forgetting. And so we found a protein called Scribble that when we knock it down and we train the flies with a single training session, these flies, the ones you can see the red trace here, they just remember way better. So the initial memory is very similar, but then they just seem to remember better. And this is just 24 hours. But when we look at very long time, even like I said, weeks, these Scribble knockdown flies, they just don't forget. They're super smart. And so by a series of different experiments, we wanted to make sure that this gene was affecting forgetting and not something else. I'm just going to show you one of those experiments. So this is what we did. So we trained the fly just the same way I told you before. So we trained the fly to associate another A with an electric shock. Then there's another B that is not associated. And then we basically reverse that contingency. Now we pair the shock with other B, so the one that was not associated with the other, and then other A is not a longer pair. And then we have two tests. One test right after the initial learning phase, and then one test after the second learning or the reversal learning. And the other difference is here that we are testing with an other A, so this, and an other C that was never experienced by the fly. So basically what this is telling us is how good is the memory for other A. And so you can see in the initial test, learning test, there's no difference between the control flies and the knockdown flies. So they do learn in the same way. But after reversal learning, now control flies, this reversal learning will induce a forgetting, a partial forgetting of the initial memory. And you can see that by this gray bar, how this memory is reduced. But when we knock down Scribble, basically there's no forgetting, there's no forgetting of the initial memory. So these flies, the knockdown flies, remember the initial memory just as well. So there is no forgetting. So just like this experiment, we did a few experiments in a similar fashion and demonstrated that knocking down Scribble, it does not enhance memory acquisition. So it's not that they're learning better. It does not enhance memory consolidation. So it's not that they learn and somehow these memories are then consolidated better into long-term memories. But it's that they actually cannot forget, so they have impaired forgetting. So we then try to understand where in the brain is this Scribble protein expressed. And so we did a bunch of behavioral assays where here we knock down. So all these bars that you see here are basically knocking down a scribble protein in different parts of the brain. Okay, and so the first one you see here, we're basically knocking down scribble in the whole brain, so all the neurons. Then we have these two drivers, so C772, 238, and R13. All of these expressed in Kenyan cells. So those Morgan body cells that I told you where we think or we know memories are encoded there. And when we knock down scribble in these cells, we have this enhanced memory. One more, when we knock down scribble in the dopaminergic neuron, so this is the tyrosine hydroxylase promoter to drive the expression of the knockdown, we do see the same effect. So when we knock down scribble in Kenyan cells and in dopaminergic neurons, we see that enhanced memory. Then we do an immunostaining, in this case with a scribble antibody, and we see how scribble is highly expressed in these Martian body cells. Not only that, when we do a gene trap, so basically we inserted a GFP tag to the endogenous scribble protein, we also see a very high expression of scribble in the Martian body. So all this data makes sense. So now the cool thing, what is scribble, right? So what's scribble doing? And so scribble, actually, this protein was described in 2000, and this was described as a very important protein for determining cell polarity during embryogenesis. So this was reported in Drosophila embryos in 2000, and they actually noticed that usually when you have a Drosophila embryo, you have a very nicely arranged epithelium that is covering the whole embryo, and you have very nice apical polarity, so all the cells are well organized. Now when you knock down the scribble, you will actually lose all of that, and the embryo, instead of looking at this very nice arranged embryo, it actually looks like a scribble. So that's why the protein is called scribble, because it looks like this arranged epithelium looks like a scribble. And that's why they call it like that. Now, scribble is expressed almost in every cell in the body. And this is a scaffolding protein. So it has 16 leucine-rich repeated domains, and it has 4 PDC domains. So it's a protein, a scaffolding protein. All of these domains are important for protein-protein interaction. And so we figure, okay, if this protein is expressed in neurons, and we know it's important for forgetting, what is scribble scaffolding in neurons? What is it doing? And so to answer that question, we basically did some mass spectrometry, right? So we took flies from that same transgenic animal that I told you, where we inserted the GFP tag on the endogenous scribble, and then have some control flies that has no that they don't have that tag, and then do IP immunoprecipitation with antibodies anti-GFP, followed by mass spectrometry. And so this is what we found. So we did this 4 times, and these are just correlations between the different biological replicates, and you can see all of them have a very nice correlation. So that means we are actually pulling scribble down. These are some of the proteins that we found that are very likely interacting with the scribble. And because Drosophila is such a good model, and there are collections for basically knocking down every gene in the genome, we can easily perform behavioral screen where we knock down all these proteins and look for changes in memory performance. So in this case, we were looking for genes that are actually increase memory performance.
Isaac, quick question.
Yeah, sure.
Are these interactors all part, or you found them or focus on them because are consistent in all your replicates of the mass spec?
There was a criteria that I took to selecting them. So
So all these guys are in all the mass spec samples you did.
Not all. There so if I selected them, if they were present in at least 75%.
Okay.
So that means 3 out of the 4 replicates.
Okay.
And we decided that because, so of course the scribble was always present, and we had a huge number of spectra in all 4 replicates. But there was an interaction with that we knew already that was true, was real.
Yeah.
And that was with RAC1. So this is a small G protein. And we actually, when we did the mass spec, we found it in 3 out of the 4 samples.
Okay.
And so that was one of the criteria we took to, even if one of the proteins was not present. Also, there's another consideration of the number of spectra. So it has to have certain number of spectra to get selected. But in that way, we came out with this list of around 60 proteins. Not all of them, we test them in the behavior because some of them were just like ribosomal proteins and things like that. So, but anyway, so we were able to screen all these proteins, and this is some published data. We're still working on that. But anyway, so now we are looking at these proteins and trying to see if they are involved in memory forgetting, and we have to follow this up with more behavioral experiments. We do have some good candidates, and we have some more data. And at the end of the talk, I'm going to show you a model of what we think is happening with this. Now, one more question we had is that, okay, so there's this debate in the field where people said, okay, what happens when we forget? Do we really erase the memory forever from our minds? Or it's just that we cannot retrieve it anymore? So the memory is there, but we cannot retrieve it. So it's like a temporal suppression of the memory recall. And so this is something that, of course, happens to everybody. We sometimes forget where our keys are, and we cannot remember at the moment. But then all of a sudden, we're doing something else, we're distracted with something, and then we remember, oh, I remember where my keys are. So this is what we call transient forgetting. But there are some memories that they just never seem to come back. You just forget and they never come back. Are these memories gone forever, or are these memories still there, but we cannot retrieve them? And so I don't know if we answer that exactly. That's a complicated question, but we did wanted to see at the cellular level what is happening with its cellular plasticity after forgetting. And so to do that, we use in vivo functional imaging. So basically, we express a calcium sensor in one of the motion body output neurons that we know it presents some plasticity after learning. So this is what we did. So this is how we do the calcium imaging. So basically, we attach the fly to a recording chamber. We open a small window in their heads, and then we put the fly under a confocal microscope, and we can actually see the GCAN signal going on. And so in this way, we actually can train the fly on the microscope. So we put the electric platform under their legs, and we can, we use some solenoids to control the olfactory stimuli. And so in this way, we can train the fly. And basically what we did is this. So we presented 2 others. So in this case, we are using MCH and octanol pre-training. Okay, so we want to know how is the response before we train them. Then we train the animal to associate, in this case, MCH with 4 electric shocks. And then after that, we recorded post-training responses to both MCH and octanol, different times after training. Okay, so now in control, so this is what we see. This is the MCH responses before learning. So you have a very huge calcium response. But then right after training, this response is completely depressed. It's gone. if you remember that model where I told you that when that node, when there is coincidence between the odor and shock gets depressed, this is the evidence for that. So you can see how the olfactory response is completely gone. But then if we wait some time and we do more recordings, then this response actually comes back. So there is a recovery of the olfactory response that correlates with the behavior. So if we want to kind of translate this to behavior, this depressed response would mean that the flies remember the task very well, and then when time passes, they start to forget. You can see how the octanol response that was not associated with the shock never changed, so it remains very high. Now, we have several ways to induce forgetting. So one thing is just wait to see the memory forgetting, or the, in this case, the olfactory response recovery. But there are some other ways that we can induce this forgetting faster. One of these ways is just to shock the flies after learning. And so what you see here, this is just a control where basically it's the same thing we saw before. We train the animal, and when we tested 5 minutes later, and so you can see the pre-response really big, post-response is gone, and then the octanol is never changed. But when we interfere this with 12 shocks right after training, you see how we recover this. And this also is observed in behavior. If we do this in behavior, flies forget about what they just learned. And we have a cellular representation of that. One more thing, you remember that reversal learning experiment? Well, here we did the same. So we train the animal with MCH and octanol as a non-pair odor, and then we reverse that. And then we have our control. You see the depression, very nice. When we do reversal learning, we did get this complete recovery of the MCH responses. And now we wanted to know what happens when we knock down, or we mess up with genes that are important for forgetting. Now, in this case, we are using this RAC. This is a multi protein that we know is important for forgetting. And basically, we are expressing a dominant negative. So we are inhibiting RAC activity. And so this is what you see. This is just a control where you see the depressed responses, and then with time they recover. When we mess up with RAC, there is no recovery at all. So this remains depressed forever, at least 30 minutes, but I think we did up to 1 hour still depressed. This response never recovered. Same thing when we do the 12 shocks interference. In the control animals, you can see how the shock stimulation recovers the MCH responses. But when we inhibit RAC, we don't see that recovery. And one more for reversal. So same thing in reversal, we see the recovery in the control, no recovery at all in the RAC flies. So this is what we think is happening. So during learning, we have these, also this would be before learning. So you see a big response when you present another, you see a big response in the output neuron, here depicted by this plus sign. During learning, you have coincidence between other and dopaminergic input that is conveying the EO signal, the shock signal, and that depresses the olfactory responses to motion body output neuron. And so when we test the memory later, you see depressed response. During forgetting, or in this case, new learning, like the reversal learning, we have these depressed responses that come from here. And then we have a new learning with a different odor. Now, odor B is the one is presented a pair with the shock. And this basically is depressing the new odor output, but is recovering or restoring the response to the initial odor A. And so this is what we think is happening. So to go back to that question, does forgetting erases the memory or not. At least from what we see here at cellular level, it seems like forgetting it's actually erasing the memory trace or the cellular plasticity. Now, I don't think that's true for everything, and of course that example I told you about the keys is a very good example of how forgetting is not just erasure; it can be a transient impairment in memory recall. Do we have time? Yeah. So one more thing, if you remember, I told you that when we knock down scribble in the dopaminergic neurons, we also have impaired forgetting. And we knew that these dopaminergic neurons are very active after learning, and they have this very high activity when they are moving and flying and active. And so we wanted to know, the hypothesis was, okay, maybe when we knock down scribble in the dopaminergic neurons, we are just doing something to this activity. And so we did more calcium imaging. In this case, we're recording from multiple dopaminergic neurons that innervate mostly the vertical lobe. So these are the PPL1 neurons, and the black trace. So each one of these is here, represented in different colors, and the black traces that you see are the control animals where we have not knockdown. And then the color trace is the when we knock down scribble. And so you can see, so for some dopaminergic neurons, we do see a decrease in this ongoing dopaminergic activity. So in this case, we see this in the alpha prime, beta alpha prime. So here the pink one, and the blue one here. Not so much in this region, the green stuff here, and then in the purple one, we don't see much because there's not much activity from the beginning. Okay, so this is our final model of how we think forgetting is happening. So after learning, these dopaminergic neurons are very active, and this activity is behaviorally dependent. So it depends on the behavioral state of the animal. If the animal is walking and is active, you have a big dopaminergic activity and dopamine release. Now we know from the studies I presented and other studies that the DAM receptor, so this DOP1R2, is important for this dopamine forgetting signal. We know scribble is scaffolding a series of different proteins. Importantly, proteins that are important for actin cytoskeleton dynamics. And we think that forgetting is scaffolding a signaling complex that is basically conveying this forgetting signal and basically reversing that cellular plasticity. I think that's it. I forgot to put my thank you slide, but just want to thank all my lab and our previous lab. A lot of what I presented today was performing at Scripps on my previous lab, especially with a lot of collaboration with Jacob Berry that you can see in all these papers. So, and yeah, just thank everybody, and thank you guys for listening.
Thank you, Isaac.
Any questions?
Yeah, we're going for questions. Do we have any questions for Isaac, you guys? Eduardo.
Yeah, hi. Thanks for the talk, Isaac. It was very interesting. So let me see if I understood from the beginning. So you have like two stimulations. One is the other, and then you have the, if I understood, whether like an electric shock, is that? So you're stimulating like in two different ways, but the neuron network is connected at some point. These two stimuli are connected at some point. The neurons are connected. And
yeah,
so my question is, so it means that if you have like the electric shock will stimulate or how do electric shock in terms of neuronal connections links the two other stimuli? I don't know if I'm clear, but I mean you have one receptor from one odorant and you have receptor from another one, and they are in the same neuron, or
No.
neurons that are different for each.
Yes, they're different. So let me go back to this. Maybe this. Actually, so the neurons that detect the odors, they're called olfactory receptor neurons. They're located in the antenna, so basically in the nose of the fly. And so those are there are many, right? And each one of those cells will express only one receptor. There are in flies there are around 60 something receptors for odorants, and each cell will express only one. They never express, well, they express two, but the other one is a co-receptor. It's not really. And so each neuron will express one. And then that information is conveyed or sent to the antenna lobe, this green stuff here. And then from there is another neuron, a different neuron it's called projection neuron. So it's very creative. So projects to the Martian body. Okay. And so when it gets to the Martian bodies are these canyon cells, and they are thousands of cells. So there are around 2,000 cells in each hemisphere. So you have 2,000 and 2,000. And so when they receive the olfactory information, only a few, like 5% of those cells will respond. Okay, so this is sparse coding of the odor. So that means that the odor representation for different odors is completely different. Somewhat, I mean, if the odor is chemically similar, well, there might be some overlap, but that's how we discriminate odors. Now, this is in flies, but this same thing happens in mammals, in humans. The olfactory system is almost the same in flies and in mammals. So only a few of these canyon cells will respond to a specific odor. Now, here I've represented that by 5 canyon cells, but just imagine that these are 2,000 really. And when when you present one odor, just a few, like 5% will respond to that odor. Now, during our training, we use 2 odors, right? One odor we paired with the shock. So that means that the flies will learn that when I smell this, I got shock. So it's important that I remember that so that I can avoid it next time. And then we use a second odorant that is not paired. It's not presented with anything. So they actually learn something about that odor too. They learn, okay, this odor is safe because I just got shock with another odor, and then I'm safe now.
Okay, yeah. So my question goes in. So first, just by curiosity, what will happen, because you do the training with one odor and the shock, and then the second odor without shock, then you inverted that. So first shock free and second shock with the, I mean, first odor free and second odor with shock. What will happen if you stimulate the 2 odors with shock, first one, then the second one, without doing in the middle the odor free? So will they still be able to discriminate, or will they respond, or they remember that the 2 odors are associated to the shock? Or
yes, they will. So basically, if you do that, then they do associate both odors with the shock. And if you test the memory in the teammates using those odors, those 2 odors, that both of them were associated with each other, then the flies don't know what to do, and so you have half of them here and half of them here, because they are more or less equally aversive. That's when you exclude the CS minus, or the other safe odor, right? Okay. When you include it, then what they remember, they remember better the most recent odor. Mm-hmm. So they will avoid more the most recent odor, the odor that was most recently paired with the shock. So the second learning part.
Right. And the other question is there any difference if you the stimulation with the first odor and the shock, and then the second odor, and then you inverted the timing in between the stimuli? if you separate a little bit more or less, will that influence how the flies respond or forget the first
Yes,
stimuli?
it does. It's very important. So timing, memory encoding is really important. So uh, if you wait between the first training session and the second one, then it's easier to induce reversal learning. So it would be easier for the flies to remember the second task, and because the initial one is going to start to decay with time by itself. So all these timings are really important. So how much time? In this case, because we really wanted to see the effect of that reversal, and we do it right away, so that we can actually see how the memory for the first pairing is being forgotten. But yeah, timing is very important.
And finally, I mean, I'm asking because I'm curious about all
No,
this
yeah.
system that you're using. But so of course these flies are they live in the lab. I mean, they're not free, but I'm wondering whether there would be odors that the fly will uh, associate more strongly with some good stimuli or bad. I mean, whether they know that there's some odor that it's very nice for them, that they like very much, and then they will forget less or more depending on the odor. So do the odor influence the response or the
Um, uh,
association?
yes, of course, there are some. I mean, that's complicated question. There are some odorants that are okay. So depending on, there are some odorants that are appetitive right from the go, right from the beginning. So they're like fruit like odorants. They flies like it. Some others, most of the others that we use are aversive, so they don't usually like it. So they are naturally aversive to them. And then there is this other complication that is the concentration of the odors. So most odors at a very or a high concentration, almost all of them are aversive. And as you decrease the concentration, that aversiveness decays. So in this case, we use two odorants that are naturally aversive from the get-go, even without mouth training. But that's why we use two, so that we can calibrate our system and they avoid them in equal manner, so that they're evenly distributed, the flies, before we train them. Now, if your question is, are they remembering some others better than others? I don't. There's not much evidence about that. I think more or less they remember in a similar way. So they have very initial good memory, at least after versal learning, and then the memory decays. Now, what we do know, and this is very interesting, is that you remember that graph when here. So aversive conditioning will produce memories that are very strong, and then they decay very fast with time. If you train the animals with an appetitive conditioning, so sugar reward, their initial memory is actually not that good. It's less than the aversive one. But then that memory, it doesn't decay, so it stays stable. And if you look at different days after learning, the memory is just stable, so it stays very, very stable. It's consolidated very easily into long-term memory. And we know when we at the beginning, when people did this observation, they were like, well, yeah, of course, this is very relevant for the animals. So they're learning where the food is, right? So they better remember it. Now we know that actually this is because the caloric content on the sugar that we use for training. So if we use sucrose or glucose or something that they can metabolize, this memory is very strong. They will remember for a very long time. But if we use a sugar that is sweet but not nutritious, like the Splenda and things like that, they learn very well, but then they forget that. So just like the first one. So it's the caloric content in the sugar that is conveying that signal that makes the flies remember very well. So that's very interesting.
Is there any other stimulus that help to that recovery other than the caloric?
Yeah, I mean, starvation is of course important for that.
The what?
Starvation, so the behavioral state is important. So if the flies are hungry, the fact that they are hungry during learning, and then you have to feed them after training. If you don't feed them after training, they won't remember. Okay, so you have to feed them. They have to get this. Yeah, that's complicated too. We are trying to understand how that happens, but yeah.
Now I'm curious. Now that so diet is so important. Have you looked what happens if you give them alcohol?
Not me, but there are some other groups that have done that. They do associate other alcohol. Alcohol is weird because it has an initial aversive memory.
A what?
An initial aversive memory, so they remember something bad. But then with time, so it's weird. With time, they actually like it. So if you look at that memory in the long term, it's actually an appetitive memory. So it's a rewarding memory, just like in humans. So alcohol has the two components, has an aversive initial stimulus that will induce an aversive memory that won't last that long. And then there is this appetitive component that is conveyed by the reward system, so dopamine. And that one will last for a long time. So they will remember for longer time. And yeah, so flies like alcohol, just like humans.
Of course.
So there's a very cool paper.
That's what I have to ask, right?
Carla Count, where she, I mean, it's not maybe not as related to memory, but it is very cool. They call, I don't remember how they call, but basically they found that if you, in a mating assay, so they put the flies to mate with females, male flies with females. And if you put the males with unreceptive females, of basically they get rejected all the time. They tend to drink more alcohol than the ones that were not rejected.
Are you serious?
Yeah, I'm serious, and that's been it's very solid results, and people have replicated this. So wow. This is something that happens. So the idea is that when they get this, I don't know how to call it, like a first kind of a frustration signal coming from the unresponsive mating, they do get this reward signal by drinking alcohol. So yeah, it's pretty cool.
Yeah, that's interesting.
Yeah, flies look more they're more similar to humans than we think.
So would you assume that mechanism of response would be something similar like your proposal here?
Yeah, I think, I mean, I don't remember the details of that paper in particular, but they are definitely the dopaminergic system that is involved in this part, so these PAM neurons, it's also involved in alcohol memories. So yeah, same circuits. Or shared circuits, I should say.
Yeah. Very interesting. Okay, do you guys have any other questions for?
I have another question just regarding the rack story. So
yeah,
because I know that the rack, it's I mean, it's involved in cytoskeletal remodeling, but also it's been found that it's associated to mitochondria and also to nuclear envelope, and it regulates not only actin dynamics, but also microtubule, somehow the direction of the microtubule sometimes. So do you have an idea where RAC will be working on in this pathway?
Well, no, really, the only thing we know is that RAC, it's not only RAC that is interacting with the scribble and important for forgetting, but also cophylline. So, and cophylline is important for actin filament depolarization. So the evidence that we have collected, not only we, other labs have collected it. Seems like it's more towards that acting dynamics, but we don't have more evidence than just the behavioral part. So we don't actually haven't seen any acting dynamics or anything like that.
Right.
Yeah.
Okay, thanks very much.
Very cool, Isaac. This is very interesting. Thank you for sharing with us.
No,
And,
thank you for the invitation.
No, it's a pleasure. So I'm gonna stop